In this set of figures, we're going to look at the factors affecting enzyme kinetics, or how an enzyme functions, what factors affect its functionality. Now, on the left here, we have what's called a Michaelis-Mitten model of enzyme kinetics, where as we increase the substrate concentration, we see that we have a rise in reaction rate. And hopefully this just makes sense, right? At a low concentration of substrate over here on the left, we have relatively few binding events with the enzyme being the little circles here, right? And because of that, we would have very few catalytic events and a very low reaction rate. Now, as the concentration of substrate rises, of course, we have more interactions. The likelihood increases of interacting with the enzyme, increasing our reaction rate. Finally, over here at a high concentration of substrate, we have the potential to reach this sort of saturation of the enzyme where all binding sites are bound by substrate. And we reach what's called Vmax, or the maximum reaction velocity. Now, what we can get from this trace is something very useful, and that is a measure of enzyme affinity for the substrate. So if we choose this sort of arbitrary reaction rate, known as 1/2 of Vmax, right? So we basically just take the Vmax and look at 1/2 of that rate. And at that rate, if we look down and see the substrate concentration required to generate that rate, we have what's called the Km, right? This is a measure of affinity because the higher the substrate concentration required, the lower the affinity would be, right? It takes more substrate to reach the same reaction rate. Hopefully that just makes sense. Now, affinity is very important, affinity of the enzyme for its substrate, but it's not the only factor affecting enzyme function. We actually have to also look at the enzyme's ability to be catalytic. In other words, what we call its substrate turnover rate down here. And that's this factor called Kcat. Right? And so really the way to determine Kcat is to saturate the enzyme with substrate over here at Vmax and just see how rapidly it can process the substrate. Right? And this varies widely between enzymes. Right? Some enzymes process a few hundred substrate molecules per second, which seems fast, but others can process hundreds of thousands or even more than 1,000,000 substrate molecules per second. Crazy fast catalytic processing of substrate. Now, ideally, right, it's these two factors together of both affinity and the turnover rate that really determine enzyme functionality. And so we combine them in this term down here, the Kcat over Km, to determine what's called the catalytic efficiency of our enzyme. And really what this is like saying is, well, what's our turnover rate given a certain degree of affinity, right? In other words, we want, ideally, for every binding event, we would get a catalytic event. And that's not always true, right? An enzyme can bind and then release a substrate without processing it. So we actually need it to stick. We need high affinity, and then we need really good catalytic efficiency. We need it to be able to process rapidly. So this Kcat over Km gives us that efficiency. If we look over here on the right, in the graph in B, we compare two different enzymes, the same axes, right? But we're comparing two enzymes. We have enzyme A here on the blue line, and we can see that it would have a high Kcat because it has very high Vmax, and it has a low Km, meaning it has high affinity. That makes this a very efficient enzyme, right? It turns over rapidly and has very high affinity, so the substrate sticks to it and is likely to be processed. In the enzyme B here on the red line, we have essentially the opposite scenario. It's much lower catalytic efficiency because its Kcat is lower, its Vmax is lower at saturating substrate concentration, and it has a much higher Km, right? And so that ratio is much lower, indicating that this would process substrate slower and have lower affinity for the substrate.